Hello, welcome to Biology, the scientific study of life. Animal skeletons function in support, protection, and movement. Types of skeleton. The three main functions of a skeleton are support, protection, and movement. Most land animals would sag from their own weight if they had no skeleton to support them. Even an animal living in water would be a formless mass with no framework to maintain its shape. In many animals, a hard skeleton provides protection for soft tissues. For example, the vertebrate skull protects the brain, and the ribs of terrestrial vertebrates form a cage around the heart, lungs, and other internal organs. Skeletons also aid in movement by giving muscles something firm to work against. There are three main types of skeletons: hydrostatic skeletons, exoskeletons, and endoskeletons. Hydrostatic skeletons. A hydrostatic skeleton consists of fluid held under pressure in a closed body compartment. This is the main type of skeleton in most cnidarians, flatworms, nematodes, and annelids. These animals control their form and movement by using muscles to change the shape of fluid-filled compartments. Among the cnidarians, for example, a hydra can elongate by closing its mouth and using contractile cells in the body wall to construct the central gastrovascular cavity, because water cannot be compressed very much. Decreasing the diameter of the cavity forces the cavity to increase in length. In planarians and other flatworms, the interstitial fluid is kept under pressure and functions as the main hydrostatic skeleton. Planarian movement results mainly from muscles in the body wall exerting localized forces against the hydrostatic skeleton. Nematodes, roundworms, hold the fluid in their body cavity, which is pseudocoelom. The fluid is under a high pressure, and contractions of longitudinal muscles result in thrashing movements. In earthworms and other annelids, the coelomic fluid functions as a hydrostatic skeleton. The coelomic cavity is divided by septa between the segments in many annelids, allowing the animal to change the shape of each segment individually, using both circular and longitudinal muscles, such annelioids. Adding to its outer edge, clams and other bivalves close their hinged shell using muscles attached to the inside of its exoskeleton. The jointed exoskeleton arthropods is a cuticle, a non-living coat secreted by the epidermis. Muscles are attached to knobs and blades of the cuticle that extend into the anterior of the body. About 30 to 50% of the cuticle consists of a chitin, a polysaccharide similar to cellulose. Fibrils of chitin are embedded in a protein matrix, forming a composite material that combines strength and flexibility. Where protection is most important, the cuticle is hardened with organic compounds that cross-link the proteins of the exoskeleton. Some crustaceans, such as lobsters, harden portions of their exoskeleton even more by adding calcium salts. In contrast, there is little cross-linking of proteins of inorganic salt deposition in places where the cuticle must be thin and flexible, such as leg joints. An arthropod must shed its exoskeleton mold and produce a larger one with each growth spurt. Endoskeleton. An endoskeleton consists of hard supporting elements such as bones buried within the soft tissues of an animal. Sponges are reinforced by hard spicules of inorganic material or by softer fibers made of protein. Echinoderms have an endoskeleton of hard plates called acicles. Acicles beneath the skin. The acicles are composite of magnesium carbonate and calcium carbonate crystals and are usually bound together by protein fibers, whereas the acicles of sea urchin are tightly bound. The acicles of sea stars are more loosely linked, allowing a sea star to change the shape of its arms. Chordates have an endoskeleton consisting of cartilage, bone, or some combination of these materials. The mammalian skeleton is built from more than 200 bones, some fused together and others connected at joints by ligaments that allow freedom of movement. Anatomists divide the vertebrate skeleton into two main parts: the axial skeleton, consisting of the skull, vertebrate, column, backbone, and rib cage. The appendicular skeleton, made up of limb bones, and the pectoral and pelvic girdles that anchor the appendage to the exoskeleton. In each appendage, several types of joint provide flexibility for body movements. Physical support on land. An engineer designing a bridge or a tall building must take into account the effects of changes in size or scale. An increase in size from a small scale model to the real things has a significant impact on building design. For example, the strength of a building support depends on its cross-sectional area, which increases with square of its diameter. In sharp contrast, the strain on that support depends on building's weight, which increases with the cube of its height or other linear dimension. In common with the structure of a bridge or building, in animal's body structure must support its size. Consequently, a large animal has very different body proportions than a small animal. If a mouse were scaled up to an elephant's size, its slender legs would buckle under its weight. In simply applying the building analogy, we might predict the size of an animal's leg bones to be directly proportional to the strain imposed by its body weight. However, our direction or our prediction would be inaccurate. Animal bodies are complex and non-rigid, and the building analogy only partly explains the relationship between body structure and support. An animal's leg size relatively to its body size is only part of the story. It turns out that body posture, the position of the legs relative to the main body, is a more important structural feature in supporting body weight. At least in mammals and bears, muscles and tendons, connective tissue, that joint muscle to bone, which hold the legs of large mammals, relatively straight and positional under the body, bear most of the load. Muscles move skeletal parts by contracting. At the cellular level, all animal movement is based on one of two basic contractile systems, both of which consume energy in moving protein strands against one another. These two systems of cell motility, one involving microtubules and the other microfilaments, Microtubules are responsible for the beating of cilia and the undulations of flagella. Microfilaments play a major role in amoeboid movement, and they are also the contractile elements of muscle cells. The action of a muscle is always to contract. Muscles can extend only passively, therefore, the ability to move parts of the body in opposite directions requires that muscles be attached to the skeleton in an antagonistic path, each member of the pair working against the other. We flex an arm, for instance, by contracting the biceps with the hinge joint of the elbow acting as the fulcrum of a lever. To extend the arm, we relax the biceps while the triceps on the opposite side contracts. To understand how a muscle contracts, we must analyze its structure. We will begin by examining the structure and mechanism of contraction of vertebrate skeletal muscle, and then turn out attention to other types of muscle. Vertebrate skeletal muscle. Vertebrate skeletal muscle, which is attached to the bones and is responsible for the movement, is characterized by hierarchy or of smaller and smaller units. A skeletal muscle consists of a bundle of long fibers running parallel to the length of the muscle. Each fiber is a single cell with multiple nuclei, reflecting its formation by the fusion of many embryonic cells. A muscle fiber is itself a bundle of smaller myofibrils arranged longitudinally. The myofibrils in turn are composite of two kinds of myofilaments: thin filaments and thick filaments. Thin filaments consists of two strands of actin and one strand of regulatory protein coiled around one another. Thick filaments are staggered arrays of myosin molecules. Notice this figure. Skeletal muscle is also called striated muscle because the regular arrangement of the myofilaments creates a pattern of light and dark bands. Each repeated unit is a sarcomere. The sacro, or the basic contractile unit of the muscle, is all the borders of the sarcomere. The Z lines are lined up in adjacent myofibrils and contribute to the striations visible with a light microscopic. Thin filaments are attached to the Z lines and project toward the center of the sarcomere, while thick filaments are centered in the sarcomere. In a muscle fiber at rest, thick and thin filaments do not overlap completely, and the area near the edge of the sarcomere where they are only thin filaments is called the I band. The A band is the broad region that corresponds to the length of the thick filaments. The thin filaments do not extend completely across sarcomere, so the H zone in the center of the A band contains only thick filaments. This arrangement of thick and thin filaments is the key to how the sarcomere and hence the whole muscle contracts. Okay, thanks.